So, in the another topic of the lymphatic organs of the immunology, today we are going to read about the thymus. So, first of all, let us discuss about the structure of the thymic globule. So, each thymic globule is surrounded by the connective tissue stroma, and it contains an outer cortex and the inner medulla. So, here is the connective tissue stroma, and it contains the outer cortex and the inner medulla, is if you can see in this picture. So the cortex contain higher concentration of the lymphocyte than the medulla, and the cortex is darkly stained because of the densely packed small lymphocyte with the heterochromatic nuclei. The outer cortex receives stem cell from the bone marrow, which divides repeatedly to form small lymphocytes. So these small maturing lymphocytes then move towards the medulla. The mature lymphocytes leave the thymus via the veins and lymphatics. So this entire process takes about 24 hours. So the medulla stands lightly. Compared to the cortex, because here the lymphocytes are less densely packed, and because of this reason, epithelio-reticular cells are more obvious uh, in the cortex than in medulla. So the medulla also contains the thymic or Husserl's corpuscle. So you can see the Husserl's corpuscle here in the medulla. So these masses are concentrically arranged type 6 epithelio-reticular cells around the central degenerated homogeneous mass. The Husserl's corpuscle contain stained pink with acid dyes, and their number increases with increasing age. So there is concentric mass if you see more closely in the microscope in the Husserl corpuscle. It is present in the medulla of the thymus. So the macrophages are found in large number in both cortex and the medulla. They engulf antigens, and they also remove the lymphocyte which might have been produced in the axis. So the location of the thymus is the anterior superior mediastinum, and the thymus is the site of T cell differentiation and the maturation. So the thymus is encapsulated, and the thymus epithelium is derived from the third pharyngeal pouch, whereas the thymic lymphocyte is derived from the mesotome. So let us move towards more clinical part of the thymus. So uh, the thymus eventually involutes by the 3 years of age, but if there is absent thymic shadow or the hypoplastic thymus, then it denotes the immunodeficient conditions in the newborns. So the immunodeficiency conditions may be severe combined immunodeficiency or Dijon syndromes. There may be other multiple immunodeficiency condition, but these two are mainly important immunodeficiency condition that may lead to the absent thymic shadow in the child. So also you have to know about thymoma. The thymoma is the neoplasm of the thymus. So the associated condition with the thymoma are myasthenia gravis, superior vena cava syndrome, pure red cell aplasia, and Good syndrome. So these four associated condition may be associated with the thymoma, which is the neoplasm of the thymus. So this concludes our topic on the thymus. So we discuss about brief about the microscopic structure of the thymus. In this microscopic structure, so in the thymic globule, the thymus is surrounded by the various connective tissue stroma, and in the thymic globule we have cortex and the medulla. The cortex is densely packed with the lymphocyte, whereas the medulla is lightly packed with the lymphocyte, and the medulla contains the Husserl's corpuscle. And we also discuss about the brief introduction of the thymus. We discuss about this location, and we discuss about the derivative of the thymus epithelium and the thymus lymphocyte, and we also discuss about the clinical condition like the absent thymic shadow or hypoplastic thymus and the thymoma. So this was for today. I hope you understood this lecture. Thank you.